So we begin our second semester with the discussion of organisms and environments. So ecology, yeah? And we're going to start our discussion with prokaryotes, the simplest kingdom, and work our way forward. So after this lesson, you should be able to describe the general characteristics of basic bacteria, list 4 ways of classifying bacteria, recognize and name the general shapes and forms of bacteria, explain the significance of cell wall structures and the gram stain, as well as describe 2 main factors that affect bacterial growth and energy. So to start off, generally we recognize 3 main groups of prokaryotes or bacteria. I'll use that term a lot just because it's a more common term, bacteria. And we also refer to these guys as germs or microbes. So our 3 main groups are cyanobacteria, archaebacteria, and eubacteria. So the bacteria basics, yeah, because all those guys had the word bacteria in it. So let's talk about bacteria basics. They're prokaryotic in their cells. They're tiny, or in fact microscopic, so we can only see them with a microscope. They're the simplest of all microbial cells, because there are microbial cells that are not in this kingdom. They're single-celled organisms with distinctive cell walls, or unique cell membranes with this thing we call a peptidoglycan layer, and peptidoglycan is a fancy biomolecule. They do not have a nucleus, as we know, and their DNA is free-floating, and it's referred to as this thing called a nucleoid. Bacteria may also have additional DNA known as plasmids that are not associated with the nucleoid, and these plasmids actually give bacteria their ability to evolve really quickly because they can exchange these plasmids with other bacteria of the same species. Kind of sexual reproduction, if you would. So bacteria, where do they live? Almost anywhere. They're capable of colonizing or inhabiting almost any environment, as long as there's water. They can be on land, in aquatic ecosystems, even in other organisms like you. Some are extremophiles, meaning they live in these extreme conditions where most life cannot be sustained. And these can be thermophiles or heat-loving organisms, halophiles or salt-loving organisms, or methanogens, methane users that use methane as their primary energy source. Bacteria grow and reproduce rapidly, actually, given the right conditions. So this actually also allows for bacteria to adapt quickly to changing environments and to have a faster rate of evolution than other organisms through mutations and gene transfer. But bacteria have 2 primary means of asexual reproduction, the first being binary fission or mitotic cell division, like you guys are familiar with, that mitosis process, or budding. So this is more like if you think of a potato that gives off a shoot and can create new fruits of itself. So some bacteria can do that also. Some bacteria actually produce endospores as their method of reproduction. So actually creating little vesicles that are identical to themselves and allowing those to shoot off into the environment. Or 2 bacteria can also join together through the process of what's called conjugation. And again, this is this very primitive form of sexual reproduction where they're exchanging those little DNA segments, those plasmid pieces, and creating new genetic makeups. So cell structure, again, they're small. They have a plasma membrane inside that other layer of peptidoglycan or the cell wall. They have no organelles, so their enzymes are attached to their cell membrane or their cytosol or their cytoplasm. They do have small ribosomes free-floating for protein production, and this circular long strand of DNA. Most have a cell wall that's usually protected by this slime coating that also aids in movement. And some have flagella, so finger-like projections that also aid in movement. Many have fimbria or pili, these little hair-like structures that line their outside. And these also help them stick together to form colonies or bands. So here you can take a closer look at the generalized structure of a prokaryote and see those different parts I just talked about. Classifying bacteria. So we can classify bacteria in 4 main ways. We can classify them by their shape, by their cell wall structure and this gram stain reaction I've been talking about, by their method of cellular respiration, as well as by the various growth factors they use in terms of energy source and nutrient source. So their shape. They can be one of four shapes, the first being cocci, or spherical. These have a low surface to volume area, meaning given their surface area or their the amount of space they take up on the outside, they have a very low volume on the inside. Examples are Streptococcus and Staphylococcus, and these are actually harmful bacteria that cause infection. So strep throat and those staph infections you hear about from the dirty water overflows. A second shape is bacilli. These are more cylindrical rod-shaped bacteria, and they actually have a larger surface to volume area than the cocci, so they have more volume given the same amount of surface area. These guys are gram-positive. We'll talk more about that. And two examples that are actually kind of harmful to humans are Bacillus anthracus, and that's associated with anthrax, as well as Clostridium, which causes botulism. A third shape would be spirillum, and these obviously, given their name, are spiral in shape, and they move in a twisting motion and usually have flagella to help with their propelling their movement. They're traditionally gram-negative. We'll talk about that. And one example that's harmful to humans would be a Treponema pallidum, or syphilis. A final shape used to categorize or classify bacteria is filamentous. So this is more fibrous. Yeah, you can tell from the picture. And these actually come in many different complex forms and are still really being investigated heavily for classification. So a second method of classifying bacteria is this cell wall structure and the gram stain reaction. So all bacteria have more than one layer protecting their contents from the environment. They have this peptidoglycan layer, which gives it rigid protective qualities. And some, however, have this additional outer membrane made of lipopolysaccharides and other macromolecules. And it's the presence of this membrane that determines this gram-positive, gram-negative thing I've been talking about. So this gram stain reaction was developed by a scientist named Hans Christian Gram, and this stain dyes gram-positive bacteria but doesn't bind to gram-negative bacteria. And again, this is likely due to the difference in their cell walls. Gram-positive's cell envelope only has two layers, a thick peptidoglycan layer and their plasma membrane. So again, they only have these two pieces, and you can look at this slide more closely in your own time and look at that structure. Gram-negatives, however, have three layers. They have that peptidoglycan layer, but it's much thinner. They also have that plasma membrane that we talked about, because all cells have that plasma membrane. But they also have this unique complex outer membrane that's got all these different fatty sugar molecules in it. And it's this unique layer that actually presents or prevents that gram stain from happening. So you can see in this picture the difference in those two membranes, right? That pink layer is that peptidoglycan. Very different. And that outer membrane is probably what prevents this stain from binding. So if we look at the gram stain, those cells are purple. And that's a positive, a gram-positive bacteria. While the gram-negatives are still red, they don't stain. A third method for classifying bacteria is cellular respiration. So this is their cellular breathing. They can either be one of three methods: aerobic, so requiring and using oxygen; anaerobic, actually not being able to tolerate oxygen at all; or facultative anaerobes, which generally use oxygen, but if there's no oxygen present, can survive without it. A final method for classifying bacteria is looking at their growth factors, the energy and nutrients they use to grow and develop. So we have two main energy sources for bacteria. So either they're chemotrophs or they're phototrophs. Chemotrophs use chemical compounds, hence chemo, and troph being how they make their food. So they use chemical compounds as their energy source. Where phototrophs use light, or photolight, as their energy source. So energy and nutrients are different. You gain your energy from your nutrients. So the nutrient source matters too. So heterotrophs tend to gain their nutrients from carbon or organic sources, while autotrophs, a second form of nutrient source, gain their carbon from inorganic sources, carbon dioxide. So this is how they form the basis of their macromolecules. So that summarizes our brief introduction to prokaryotes. I hope you look through it again and find a little bit more information for yourselves the second time around. Good luck answering your questions.